Silva de Leonardi. So just for a quick run of show, for those of you who aren't familiar, I'll give just a quick introduction to Noah. And then we have the public portion of his defense. So he'll give a talk for about 45 minutes. There'll be time for questions from the audience and from Zoom. And then his committee and Noah will retire to a private room and talk more about the dissertation. Right, so a little bit about Noah. Noah was born and raised in Las Cruces, New Mexico. He did his undergraduate in mathematics at University of New Mexico. And then he returned for a bachelor in biology. And when I recently asked him why this was, he said that he explained to his family at the time that he did not want to be a nerd. And I'm so pleased, like the best part of our job, right, is to like help people achieve their career goals. So I'm so pleased that I was able to help you with that. Sort of after getting the training in mathematics and biology, Noah found what I think is one of the most interesting intersections of math and biology, which is epidemic dynamics. And so he did a master's with Helen Wearing at UNM on modeling epidemic dynamics of dengue fever. And then he joined our group here in 2017. And at the time, I thought that the most important thing for each person in my group was to be working independently, leading a really hard problem. And so I said, I think we had a conversation about, you know, infectious disease theory kind of gets it wrong really badly for all social animals. And I said, no, how about could you please fix that? And this sort of brings me to one of my favorite traits about Noah, which is that he is sort of relentlessly cheerful, which is a wonderful thing, as we all know, to have in a colleague. And so he relentlessly, cheerfully worked on this problem and achieved results that I'm really excited about. They have really clarified my thinking about the issue, and I'm really confident that's going to be true for others in the field as well. And so it's truly been a privilege to work with Noah on these problems, and that you're going to hear about in a moment. In terms of next steps, Noah is going to spend, I think, more time with his baby, who was born the day after he submitted his dissertation. So that's exciting. And I think that is where I leave it. And so Noah's talk is entitled Epidemic Dynamics When Superspreading Occurs in Bursts, and take it away. So I think everyone is here except for David Hendricks and Yevgeny, and David Hendricks is online. Do you know if the committee is actually required to be here for the whole public? Yeah. Is he accepted the calendar, and hopefully he is.
It goes in the front one. That's okay. Hi, can you hear me? Everything's okay? Great, thank you. Thank you, Ben, for that introduction. And like Ben said, my talk today is called Epidemic Dynamics When Superspreading Occurs in Bursts. And today I'm going to talk about a potentially useful quantitative framework for thinking about epidemics, specifically in social populations, where they might collectively amplify or suppress a pathogen. To start with, I'm just going to give a brief overview on epidemic dynamics and assortativity, which is a feature of social populations. And by epidemic dynamics, what I mean is patterns of disease spread. And so what do I mean by patterns of disease spread? Well, to start out with, today I'm going to be talking about diseases that exhibit this thing called direct transmission, though diseases can spread in many different ways. In direct transmission diseases, what has to occur is that there has to be some kind of infectious contact between two individuals, one individual who is infected with the pathogen and one individual who is susceptible to the pathogen. Examples of this include SARS-CoV-2, as you probably know, influenza, measles, and essentially where you can get sick by being near someone else who is sick. At the population level, this infectious contact can happen multiple times until either the infectious individual recovers or there are no more infections at all. Then the same thing happens for secondary infections. They infect individuals, their infections infect more individuals, and eventually either everyone in the population is infected and there are no susceptibles left, or the infection dies out because everyone in a particular generation produces no infections. Because of how this disease can be traced from person to person, it's really natural to think about these as a branching tree process. Essentially, you can retrace who infected whom and build a mathematical and visual representation of how a pathogen has moved through a population, like the one here. Branching processes and the mathematical theory behind them are really important to disease ecologists. They represent the way that we think about parent-child relationships, and they're found throughout biology in things like phylogenies, river stream networks, as well as family lineages. And they were actually the first investigation into branching tree processes and the first description of them was talking about familial relationships, specifically the extinction of surnames. And what that means is that the language behind them has a lot of kind of family language. And so I'm going to be using these terms like parent and child, which are representing different generations or strata of relationships between individuals. But we can also use branching processes to represent outbreaks of infectious disease. But in this case, if I say child or children, what I'm talking about is the individual or group of individuals who have acquired an infection from any particular quote unquote parent infection. In these cases, I am not necessarily talking about transmission assortativity, though for any of the parents in the room, they know that sometimes it's actually the other way, where the kid gives the disease to the parent. And so I also want to I think it's really important to note that individuals involved in transmission and particular super spreading events are most often the impacted parties of these things, not the perpetrators. This is true for both parent and children relationships, and it's something that is not always treated with sufficient empathy in our conversations. Someone's not a super spreader. A super spreading event has occurred. For infectious disease branching processes, they start with some kind of patient zero, and this just represents the first infection in a particular context. This context could be the first infection in a population, the first infection in a particular group of individuals, or the first infection in a species. When a transmission event occurs, we can represent this as a line or an edge on this graph. And these lines connect these parent-child relationships or infector-infectee pairs and show how the disease moves through a population. Then we can visualize the whole structure of the outbreak, and this can characterize how an outbreak progresses. So to kind of sum it up, transmission trees, which this is an example of, is a branching process that's been applied to infectious disease spread. A really important part about disease dynamics and epidemic dynamics is that not every single individual infects the same number of other individuals. Infectors will have a greater or fewer number of infectees depending on the number of infectious contacts that they might have. And what this represents is this thing called transmission heterogeneity. And transmission heterogeneity is just the variation in the number of infections caused by any given infectious individual. Occasionally, we'll see this heterogeneity to an extreme, and we'll have a greater than expected number of children acquiring their infection from a particular parent. And so we call this super spreading dynamics. And you've probably heard of super spreading dynamics with the COVID-19 pandemic. A key result in disease ecology is that super spreading changes outbreak dynamics by making them more explosive, but also less likely. Diseases that exhibit super spreading can have 80% or more of infections caused by the top 20% of the most infectious individuals. And this is because while some infectious individuals produce many infections in these types of dynamics, most individuals actually cause very few or no children infection as we go through. So you can see in this graph by Lloyd Smith et al. That we have some of these diseases that are causing 80% or more of the infections caused by the top 20%. The y-axis shows the proportion of transmission due to the most infectious 20% of cases. And then the x-axis here shows how they measured transmission heterogeneity in a population, specifically this parameter called the dispersion parameter, K. And this K is a little bit counterintuitive because it is actually inversely related to heterogeneity. When K is low, or here below 1, down into the 0.01, this means a very highly heterogeneous outbreak, versus when K is high, this means a more homogeneous outbreak where less superspreading dynamics are occurred. We can also see on this graph that there's a lot of pathogens of interest that follow this same pattern, like SARS, the first one. This was made before SARS-2 was here, as well as measles and monkeypox. Their research actually went further to show the importance of transmission heterogeneity in outbreak forecasting and in outbreak control. On the left-hand figure, we can see their results regarding that idea that outbreaks are more explosive but less likely. So here we have the dispersion parameter on the y-axis, and then the first generation with 100 cases on the x-axis. In the most heterogeneous populations or simulations that they did, what they saw was that these simulations would reach 100 cases in a generation very quickly. So their first generation was in the first 10, but only a few of those cases of those simulations were actually reaching that threshold. Versus in a more homogeneous, it takes longer to get to that 100 cases in a generation threshold. However, a majority of their simulations were reaching that threshold. On the control side, what they found was that especially in heterogeneous populations, targeted control, where you're targeting the most infectious individuals, is much more effective than in populations that are more homogeneous. And this is a really important result, but essentially it's saying that focusing resources on targeting superspreading is most important when superspreading occurs in a population, which, is kind of a tautology. However, it is a really important result, I promise. One thing that is not changed by transmission heterogeneity is the critical threshold of epidemic dynamics. One big question that we have in disease ecology is the probability that a relatively small local outbreak will scale up and become a full-blown epidemic. When an outbreak scales up and becomes this full-blown epidemic, we say that it's exhibiting supercritical behavior. And conversely, when an outbreak that does not scale up, a small local outbreak does not scale up, we call this subcritical behavior. In the classic theory, whether or not an outbreak goes supercritical is determined by the reproductive number R, which I will define in just a moment. The critical threshold is the dividing line between that subcritical and supercritical behavior, and it occurs when that parameter R is equal to 1. By definition, R, the reproductive number, is the expected number of individuals or infections that a randomly selected individual in the population will generate. Sometimes you might see this represented as R0, which is the basic reproductive number, which is just a special case of R when the entire population is susceptible. Like I said, this R determines the critical threshold, and when R is below 1, no major outbreaks can occur. But when R is above 1, a major outbreak may or may not occur. And so to review, transmission heterogeneity is really important in outbreak dynamics, but increasing or decreasing heterogeneity does not change that critical threshold. It only changes how the outbreak is structured, and R equal to 1 being the critical threshold still remains. And so notice, we're just thinking about R here. We don't actually consider K when we're thinking about critical threshold in the classic theory. And basically what I see this as classic theory telling us is that social structure does not matter in determining the epidemic threshold. And that seems pretty unlikely. If classical theory is telling us that heterogeneity doesn't matter, maybe the reason that it doesn't matter is because of how we're thinking about heterogeneity in the theory as a whole. So let's look at some features of social populations that we do know affect epidemic dynamics and might affect epidemic thresholds. In social populations, transmission heterogeneity might not be enough to model these epidemics, especially because social populations, like humans, exhibit this really interesting phenomenon called assortative mixing, which is essentially the tendency for birds of a feather to flock together. Like individuals are going to associate with other like individuals more than they would the mean individual in the population. Here, we can see this phenomenon in the publishing habits of academics, specifically network scientists, because this is from network theory. Individuals from this discipline are more likely to publish together if they are close geographically or located at the same distribution, and you can actually kind of group them into these different groups. An example of assortativity in epidemic dynamics is measles epidemics in vaccine-hesitant populations. And so this is just kind of showing some of the outbreak sizes of measles in Canada in the late 1990s and 2000s. And I see this as an example of transmission assortativity. And I'm going to define transmission assortativity right now. But first, note that transmission assortativity and transmission heterogeneity are different. It is one thing to have a population where you have a small proportion that's unvaccinated and therefore has a different transmission potential. It's another thing for those individuals to form an insular subpopulation, which is an example of the assortativity. And so thus I define transmission assortativity as the tendency for infectious individuals to infect others with similar transmission characteristics. And two fundamental factors make this really important in social populations. First, there is predictable biological variation in transmission potential. An example of this is age-varying transmission. Individuals of similar ages are more likely to infect having the same transmission characteristics as individuals with similar ages as opposed to other ages. The second, we know that in social populations, individuals are actually going to assort on these self-same characteristics. For example, age assortative mixing. Individuals of similar ages interact more than individuals of different ages. And the point is that the same factors that individuals are assorting on are the same factors that determine transmission potential. And so this kind of brings me to the first question that I addressed in my thesis, which was, does transmission assortativity change epidemic dynamics? And I also examined the critical threshold in that. If you want to know more information about the math, please don't hesitate to reach out afterwards. I would love to talk about it, but I'm just going to give a brief overview of the methods here. So I based my model on the branching process model by Lloyd Smith and colleagues, from which those figures earlier were generated. And I incorporated transmission assortativity by allowing autocorrelation in the transmission potential of infector-infectee pairs. But I really want you to focus on the three key parameters that I was using in my model. The first parameter is K, which is that same measure of heterogeneity we saw earlier. And just a reminder that when K is small or less than 1, we have a heterogeneous outbreak, versus when K approaches infinity or gets large, we have a more homogeneous outbreak. The second parameter is called nu bar, which I call the base individual reproductive number. For now, just think of this as equivalent to R. It's the expected number of infections an individual will cause. Delta is my new measure, and it's a measure of my new parameter, and it's a measure of transmission assortativity. When it's 0, there is no transmission assortativity, and the Lloyd-Smith model is recovered as a special case. But as it increases and becomes bigger than 0, some transmission assortativity is present, and we enter into dynamics that thus far have not been explored. So just to recap, 3 key parameters that I'm going to be talking about. We have K, which is the measure of transmission assortativity, nu bar, which is this V with a hat, base individual reproductive number, as well as delta, the measure of transmission heterogeneity, or transmission assortativity. To test its importance, I approached modeling transmission assortativity in 2 different ways. First, I'm going to show you how a transmission assortativity changes outbreak behavior by simulation in branching process models. I'm going to demonstrate how epidemic dynamics change when transmission assortativity is present, as well as show evidence that transmission assortativity is a critical measure in the is an important measure in the critical threshold of a disease. In the second model, I'm going to show an analytical relationship between the critical threshold and transmission assortativity, and actually all of the parameters of my model. And I'm going to demonstrate how transmission assortativity can be used to evaluate vaccine program efficacy. But first, I want to give you a little bit of intuition about how transmission assortativity changes epidemic dynamics. let's start at this bottom with this first figure. So we have, in this case, we have low heterogeneity, because k is equal to 1. So, it's not below 1, it's at 1. And we have no assortativity. So this is kind of the base model. It's the geometric model if you are interested. And just also to note, there's a black dot here. It might be a little bit hard to see right here. That indicates the first infection in this tree. When we have low heterogeneity and no assortativity, we get this spindly look to a tree. It's a tree that's kind of spindly. Most individuals are infecting the average number of individuals. There's not really any super spreading dynamics occurring. Kind of shifting over to the bottom right here, when we have high heterogeneity, so we have that black dot here, but we still have no assortativity, we get a series of bursts that happen with a lot of die-outs coming off of those bursts. This gives a bushier look to the tree. It's a more bushy tree. And then finally, here's that first infection here. When we have a medium amount of heterogeneity and some assortativity, we see that we get both types of behavior happening in the same tree. Near the initial infection, we're really close to this kind of spindly nature of the tree. But as individuals kind of become more and more super spready, it gets bushier and bushier, and that behavior kind of shows this thing in epidemic dynamics that we call smolder and bursts, which are pretty difficult to represent without transmission assortativity. And just so you know, this is the only time you're going to see living creatures in my talk. These two trees that may or may not be living, to be honest. That one looks pretty alive. Okay, so now turning to some of my results simulating outbreaks with my new model. The first result that I'm going to give you, I'm going to orient you to the graph first. Along the y-axis for both of these graphs is the probability that an outbreak is of a specific size. Along the x-axis is that size, it's that size of the outbreak. On the left, I have only varied the amount of transmission assortativity, and on the right, I've only varied the amount of transmission heterogeneity. For both graphs, new bar is 0.8, which should be below the critical threshold when there's no transmission assortativity present. And on the left here, there's a moderate amount of super spreading. We have a k equal to 0.2. So let's start on the left side. What we see is that as we increase transmission assortativity, this delta parameter, up not very far, right? We're going from 0 to 0.06. We increase the potential size of outbreaks, and eventually start getting outbreaks that are of a size that we know that they will become self-sustaining, and so we no longer simulate them, which is kind of this edge of this graph, is that they've become self-sustaining, they become supercritical. On the right-hand side, what we can see is that this behavior that's occurring on the left-hand side is because of transmission assortativity alone. While transmission heterogeneity does have an impact on the final possible size of outbreaks, a whole order of magnitude difference, that's pretty big, we aren't seeing it causing these outbreaks to become self-sustaining and thus hit that critical threshold. And so the key takeaway here is that when transmission assortativity is increased, all else being equal, it increases the chance of a major outbreak occurring, because it can make a huge difference in the size of the outbreak. Increased transmission assortativity, major outbreaks are now possible with just a small one. An additional result is I found a way to start visualizing this critical threshold for various parameter sets. In the critical theory, which corresponds to the bottom border of these graphs, in the classical theory, not the critical theory. The classical theory, which is the bottom border of these graphs, the critical threshold is at nu bar equal to 1. So you can see here, nu bar equal to 1 for all three of these graphs. But as we see, when both transmission heterogeneity, that k going from infinite down to 0.2, as well as transmission assortativity, which is the y-axis here, increases, we see that this critical threshold bends away from 1 and means that outbreaks exhibiting transmission assortativity can have major outbreaks even when this nu bar is less than 1, which is to say that social populations are really good at making things go viral. They can do it even when classical theory says maybe they shouldn't be able to. And so to further explore this change in the critical threshold, I had to change model types. Branching processes are really good for emergence and elimination scenarios or looking at a specific outbreak, but I wanted to look at more long-term behavior, and this subsequently led me to use compartmental models. In the compartment model framework, I can explore the critical threshold using this thing called the next generation matrix method. And this essentially solves for that basic reproductive number R naught when there's no infections in the population. And so to accomplish this, I embedded a discretized version of that branching process model that I gleaned the results from before into the connectivity between the subpopulations of this compartment model. But I just want to remind you before I get into some results, I know that was a little bit technical, that critical threshold of R being greater than 1, that's when supercritical behavior can occur. And when R is less than 1, that's when subcritical behavior occurs. So what I found when analyzing this separate framework was that this critical threshold is a function of all 3 model parameters. Nu bar, which for these simulations here are all set at nu bar equal to 1, the transmission heterogeneity K, and transmission assortativity delta. To orient you to this graph, the y-axis here is transmission assortativity from 0 to 0.25. The transmission heterogeneity is from 0 to 2, or from 0.01 to 2. For each of the scenarios shown, areas where R naught, or we would expect supercritical behavior in the system, are represented by the lighter colors, whereas the darker colors represent when R is still at 1. And what we see is that especially when there's some transmission heterogeneity present, if we recall, there's a lot of diseases that have transmission heterogeneity under 1, even a modest amount of superspreading, where we come into this area right here, a modest amount of transmission assortativity, where we come into this area here, can make outbreaks, major outbreaks, possible. So not only does my research show that transmission assortativity matters, it also shows that transmission heterogeneity is a factor in determining critical thresholds. We just needed a different framework to think about heterogeneity and how it might be structured. Because R naught was being affected by transmission assortativity, this kind of made me think that it might have major impacts on control strategy, and I expect it to have major impacts on control strategy. To simulate this, I used a similar model where some individuals were perfectly vaccinated, which means that instead of having to go through the whole process, they went straight from the susceptible compartment to the recovered compartment. And recall earlier that I said that targeted vaccinations worked best for highly heterogeneous populations. And so here I actually applied a targeted vaccination on a population for a disease where the R naught was going to be 3. I separated this population into the top 20% by transmission potential, like we saw before, and the bottom 80% by transmission potential. And then I targeted vaccination of that top 20%, vaccinated 95% of those individuals versus only 70% of the rest of the individuals, for a total vaccination fraction of 75%. And that was a lot of percentages in a row. There's only one more left. To just give you a result from classical theory, there is this thing called the critical vaccination fraction, which is the percentage of the population that you need to vaccinate to prevent a major outbreak from occurring. Since I told you that the R naught is equal to 3, I can tell you that the critical vaccination fraction here is 66% because it is 1 minus 1 over R naught. And I promise that's the most equations that I have in my talk, in this version of my talk. Okay, so then I applied that to the same kind of system that we saw before. And what we find is that targeted control can fail when transmission assortativity is present. So this graph shows the RVax, which is the R of the system when this vaccination program has been implemented. And then I subtracted 1 just to show where it was actually suppressing and becoming effective in this blue area here, versus the vaccination program being ineffective, which is represented by this red area here. On the y-axis, we still have our measure of transmission heterogeneity. And on the x-axis, transmission assortativity, they're hard, sorry. And on the x-axis, we have our measure of transmission heterogeneity. What we can see is specifically in this swooped area here in the middle, is that small changes can have big effects. For instance, small changes in assortativity, which represent going up and down on this graph, can lead you from an area of effective control to ineffective control with pretty small motions. Note the y-axis here. The same thing can actually be said for small changes in transmission heterogeneity. In this case, when we go left to right, we can see that we go from an effective to ineffective to effective as we change the amount of heterogeneity. And this is a really major implication for how vaccination programs might be designed in the future. Okay. To recap the effects of transmission heterogeneity before I go into a small vignette for my second results, I demonstrated that it can change outbreak dynamics. It can show that smolder and burst behavior that we talked about. It is a factor in determining the critical threshold for an epidemic, with outbreaks showing higher transmission potential, higher transmission assortativity, having a higher potential to become full-blown epidemics. We said that this is distinct from transmission heterogeneity because heterogeneity alone does not change that critical threshold. Finally, we know that transmission assortativity has major implications on control policy. We might, if we do not consider transmission assortativity in our design of our control programs, we might end up in a case where the traditional measure of how many people to vaccinate might fail to fully protect the population. But all of this begs the question, can we measure transmission assortativity outside of theoretical contexts, or is it cryptic and not detectable in data that might be gathered by epidemiological investigation? Which leads me to my second question, and I'm going to show you how I did measure transmission assortativity in outbreak reconstructions, and I'll show you why. So to answer this question, I had three research goals that I went through. The first was to construct a hierarchical estimation approach, and to do this, I used a Bayesian framework in the STAN programming language. I validated this estimation method with synthetic data, and specifically synthetic data that might be gleaned by an epidemiologist doing case and contact tracing. And then finally, once I was satisfied with the performance of this model, I applied this hierarchical estimation approach to empirical transmission trees from the literature to demonstrate how we might estimate transmission assortativity in real-world outbreaks. In the interest of brevity, I'm only going to talk about this last goal, applying the hierarchical estimation approach to empirical transmission trees taken from the literature, even though I will say that STAN code looks really good on a presentation. It's just lines of text. Everyone loves a line of text. So the transmission trees that I analyzed represent three different diseases in 5 different contexts. The first disease analyzed was COVID-19 or SARS-CoV-2 in Xi'an, South Korea, as well as Beijing, China. I also analyzed 2 Ebola outbreaks, one in Conakry, Guinea, and one in the Gulu district of Uganda. And finally, I analyzed a SARS outbreak in Beijing, China from the early 2000s. And before I show you the results of our estimation methods, just remember that in our theoretical results, we saw that when delta just got increased a very small amount, right, going from 0 to 0.06, we saw really large and fundamental shifts in outbreak behavior from subcritical to supercritical. And so what did we find? Well, we found that transmission assortativity can be found at sufficient levels to change outbreak structure. This figure shows the posterior distributions of the estimation of delta for the 5 outbreaks that I analyzed. The y-axis shows the different outbreaks that I analyzed, and the x-axis shows the value of delta. I have this dotted line, which is just indicating that delta equal to 0.05, which we remember from this figure should be sufficient to change outbreak structure. And note here that 2 outbreaks, we didn't find really much evidence that delta is playing a role, that transmission assortativity is present. It's pretty low levels. However, in 3 outbreaks, the COVID-19 outbreak in Beijing, and both Ebola outbreaks, we do find high levels of transmission assortativity that should be able to change them. And what this means is that we can detect transmission assortativity in this real world data. And I think that outbreaks are offering a glimpse into the underlying social structure of the populations that we analyze them. And to just kind of talk a little bit more and kind of do a small interpretation, if you will. I'm going to talk about two specific outbreaks, the COVID-19 outbreaks in Xiaonan and in Beijing. While both of these outbreaks were of the same disease, they occurred in different contexts, which may have affected the level of transmission assortativity seen within them. In Xiaonan, this was the first major outbreak in the area, and the first case was in February 15th, and control measures didn't start until later on, February 23rd. This means that the entire population was probably susceptible, and this could indicate a time where only superspreading was occurring and not much transmission assortativity. On the other hand, in Beijing, this happened later on in 2020, in June, which was under zero COVID policy in China. This means that control measures were in place, and since it was later, we might not know the susceptibility landscape. And this is really exciting to see that it actually varies for the same disease in different contexts. But it also suggests a counterintuitive result. And this counterintuitive result is that transmission assortativity might increase when control measures are implemented, which would in turn reduce the efficacy of those control measures, like I showed in that red and blue graph of the vaccination program. This would mean that we might end up in a tragedy of the common situation, like those studied by my colleague Brian Lin, where the increase in transmission assortativity and assortativity of high transmission individuals might eventually overcome the measures that are put in place and lead to large-scale outbreaks, which that seems like it matters, right? That seems like it might be important. So, like I said, transmission assortativity, I can say it exists in empirical outbreaks, and it can be detected and quantified at levels that will fundamentally change outbreak structure. And I see this as a very important step to taking transmission assortativity out of the theoretical realm and making this parameter useful for policymakers and other research scientists. And with that, I'm going to make just a few concluding remarks. The first is to say that transmission assortativity matters. It changes outbreak structure, and it needs to be considered by other disease researchers. As we prepare for pandemics of the future, we really need to be ready to characterize these outbreaks of infectious disease in the best way we can with the most tools we can. And to this end, I think that neglecting transmission assortativity could lead to inaccurate forecasts, as well as potentially ineffective control measures. And that's really important. The next, I would say that we need to continue to measure transmission assortativity and to better understand its contributing factors. Outbreak reconstruction is getting more precise and modern, and we're getting more of them, which means that more outbreaks can actually be analyzed in the same way that I did. And I also anticipate that other measures of transmission assortativity can be proposed and can be acceptable. As researchers gather more information about the other factors that influence transmission assortativity in a population, we can look to the future of predicting transmission assortativity before an outbreak occurs, and then designing control measures that can leverage the fact that it has a crucial role in epidemic dynamics. And with that, I'd like to make a couple of acknowledgements on my talk. How are we doing? Oh, great. First, I'd like to thank my committee, Ben, Mark, Yevgeny, Dave, and Jim, as well as the IB front office, Tara, Tracy, and Melissa. I couldn't do this without a committee, and IB couldn't exist without the front office. Next, I'd really like to acknowledge the DL Lab, Ben, Rachel, Isaac, Justin, and Liz, past member Liz. You know, science is a community, and I'm really grateful to be part of this lab, which is doing such great science. I'd also really like to thank the Bi22X teaching team, Lori and Carmen. Really great mentorship and opportunities for growth that you've offered me, and also the prep staff as well. The prep staff and my TA colleagues, you know, it really takes a team to teach all of these undergrads that we teach, and I really am thankful for the opportunity to teach alongside all of you. Next, I'd like to acknowledge my friends that I've had throughout my academic career. Just to name a few of them, I'm sure there are people I've forgotten, I'm sorry. Michael and Larissa, from lab beers in New Mexico to our current board game nights, being able to chat with other, you know, disease ecologists from different levels has always been great. The Eugene rugby and floating crew, it's nice to get away from academia every once in a while, and rugby is my outlet. Thanks again to Carmen and my other Eugene friends, Becca, Eric, as well as Tim. To my former roommate, Michael, he, was with me for a really long time at the beginning of my PhD, and he's really doing some really awesome science. And then finally, Silke Bachhuber. Really thankful for Silke. She does so much for me and my family. Sorry. I really couldn't have done it without all these people. Really thankful for my family, Mike and Debbie, my in-laws. Thanks especially to Debbie, who's currently at home with my wife and my new baby, making sure that he's okay. My parents, I couldn't be there without. They raised me to be the person that I am today. The reason I value education, as well as my sister and my brother-in-law, my sister Julia Silva and my brother-in-law Colin. They've always been there for me. It's really great to have a family that really cares about what I'm doing. And then finally, I'd like to thank my best friend, my lovely wife, and of course, the mother of my new baby boy, Kate. I love you with all my heart. And you've supported me through some really tough times in graduate school. And I can't thank you enough. Finally, the last acknowledgement I have is for the land which Oregon State University is located. I think it's important to acknowledge the history of how Oregon State University came to be and acknowledge the people displaced. I also think it's important to support those people in any ways possible. And to that end, I ask you to join me in donating to the community of the Siletz Tribal Art Society, which is part of the Confederated Tribes of the Siletz Indians, as well as the Tumwata Village Redevelopment Project, which is a project from the Confederated Tribes of the Grand Ronde. And both of these are kind of projects that are run by tribal members to help them recover and maintain their heritage. And with that, I can take any questions y'all might have.
Noah, questions from the audience?
Social factors control transmission sortativity, like COVID-19. Okay, so I think this is one of those things that's common. Reality is such a big function where if we're thinking about designing control strategies, the control strategies kind of revolve around things that it's group has been social creatures to do, which is social creatures are going to gather in this way. Action. Oh no, it's okay. Yeah, so they're going to have different levels of sociality, and asking them to not gather is pretty rude, but it might be one of those things. Biological, I think that the biological things are things that are still being explored. There are theories about infectious individuals being more susceptible to disease as well, and so like the interaction between the immune system and how many secondary infections a particular individual can cause are really complicated. And I think that more work will be done on that, and we'll have a better idea of how we can actually measure transmission heterogeneity in individuals. And once we know that, we can then take the next step to think about how we might design communities that can limit the assortativity of those individuals. Okay, yeah, so it's a question about the timescale of the, sorry, I'm just saying it from the mic, of these outbreaks and assortativity. So most of these outbreaks were short because I actually do not know the exact number of days, but I know for most of them, like at least the one in Xiaonan, that was literally like maybe 2 weeks of case contact, because as you extend time, contact tracing becomes harder and harder and harder. Epidemiologists, the boots on the ground especially, are doing one of the hardest jobs, right? Where they're trying to actually figure out how disease spread. I think the longest one was probably the biggest one there, with the this outbreak in Beijing. I think this was on the order of like maybe like a month or so. And to answer your question a little bit more though, it is time varying and it's also behavior varying. And so it is, to estimate it, it is a snapshot at any given time, because if you implement more control strategies, you're probably changing transmission assortativity. If you change the susceptibility landscape and you actually run out of those high transmission individuals, you're also probably changing the disease landscape. So you were very right that it's probably a snapshot at any given time, but I still think that we could leverage it, especially for control strategies, because we do know that some diseases transmit similarly to others, so we could possibly use past outbreaks as instructions for the future.
Question says, is there a relationship between absorptivity and a population density? Most of modern recreation were large human populations.
Yeah, so the question is asking, is there a relationship between transmission assortativity and population density, talking probably about like a city with multiple different subpopulations in that city where some are more dense than others, but they're all richly connected. Maybe not. That's one of those cases where density is a predictor of transmission potential. If you are more tightly packed, then you will actually have more density. And your transmission potential might be higher. But how those are interacting, I mean, I think I first actually got both me and Ben kind of first got the idea about this at looking at like census data of connectivity between different populations. And one of the things that you can see is that dense populations do often interact with other dense populations. And so the assortativity itself is about the connectivity between those dense populations, not about the fact that their transmission potential varies because of density. And so maybe I would actually say that density is more of a factor in transmission heterogeneity, whereas transmission assortativity is about the connectivity.
All right. Well, let's take a chance to thank Noah for a great presentation.
Thank you all so much.